So let's welcome Dr. Somnath Ganguly to the virtual stage for leading this session on Decoding Impurities, a comprehensive guide to regulatory standards.
Okay. Okay. So, good morning, everyone. First of all, let me thanks Eminence Business Media for the inviting me for the third impurity conclave. And incidentally, I am present in the last two also. So, thank you again, Gunit and the team, entire team. Okay, to start with, as Neeraj has already explained that it is a decoding of impurity. Whatever I am trying to say, the presentation focus will be on the specifically identification, reporting, and qualification of different impurities to get the controlling the specification. And I am mainly focusing on the ICH guidelines and on the ICH guideline only on this part because otherwise these guidelines are very vast and the time is too short and Gunit and team is very punctual on time as I am also. So anyway, we are giving basically in the entire 2 days I have seen there are a lot of interesting topics are there. So I have thought of to recapitulate all the things to make everyone on the same stage, same page. And these majorly most of the things are known, but just to I am making it for everyone in on the same page maybe. Okay, so I'll cover the organic impurities, genotoxic impurities, and the elemental impurities. So guidelines, I am not go through the entire line by line. The ICH guidelines are applicable for all regulated and semi-regulated markets, but as we all know that the different regulatory agencies are, it is not mandatory for them to follow ICH guideline because country-specific guidelines are also there with respect to impurity. Okay, and these are all known to everyone that Q3A for the drug substances, Q3B for product, Q3C for residual solvent, ICH-M7 for mutagenic genotoxic impurities, and Q3D for the elemental impurities. Today, I am trying to focus on salient features of Q3AB, M7, and Q3D. Now, these are some definitions that we all know. So I am not reading all these things. Impurity profile, what is the identified and unidentified impurities present in the drug substances, specified impurity, unidentified, identified impurity, unidentified impurity. I am not going through the definitions. It is known to everyone. Unspecified, genotoxic, and elemental impurities. Because in the during presentation, these terms will come off and on. Now, this is one snapshot of types and sources of impurities. This is not prepared by me because it is I got informed one my close friend is in recently published in LinkedIn, so I have got it from there. But it's a very nice in a one shot. Impurity is one that all we know: organic, solvent, and inorganic. Organic impurity could be once again degradation impurity and the process related impurity. Solvents are everything. It could be organic, mainly organic, or inorganic liquids are also there. And that inorganics reagent, heavy metals, inorganic salt, etc. And the degradation fates are with all degradation related impurity, maybe the API degradation, API excipient interaction, API residue interaction, API container interaction in the formulation. And excipient interaction, starting materials, byproducts, intermediate, these are of API, reagent ligands in the API process are all the process related impurities. Okay, this snapshots when I think the IBM team will share you the presentation, but this is I like this snapshot very good. It may help for anyone. Now come to the organic impurity. So it's a very old term, reporting threshold, identification threshold, and the qualification threshold. So we all know all these things. Once again, I am not putting much time on that. I'll try to explain the reporting, identification, and qualification threshold with some examples. This is all from the ICH guideline itself. that if the maximum dose is greater than 2 gram per day or less than 2 gram per day, these reporting thresholds, identification threshold, and the qualification threshold, the values will change on, will keep on changing. So let us come first to some of the actual illustration, and my entire focus will be this calculation of specification, etc. Okay, because I feel in many times people are moving on this and moving in circles, what is the right things or not. So say an API X have the maximum daily dose of that formulation in 450 mg. So it is greater than, which is surely less than 2 gram per day. So what will be the reporting threshold is 0.5%. So how we will calculate the this thing, say for or identification threshold. I have given one example. Suppose identification threshold is 0.10. So calculation is like this. Okay, 1 milligram is how much of 450 mg? It's a very arithmetic. It is 0.22%. So which is more than 0.10% as per the guideline, which is lower. So in this case, my identification threshold will be 0.1, not 0.22. Similarly, qualification threshold also 0.15, not 0.22. So it is best every time we have to calculate and we have to fix. Okay. Now it is with respect to the drug product, understanding threshold for drug product. So it here it is once again less than 1 gram and more than 1 gram. So reporting threshold is 0.1%, or if it is greater than 1 gram, it is 0.05%. So again, if it is suppose one product, say metformin drug product, maybe 2000 mg is the maximum daily dose. This is more than 1 gram category, the limit will be 0.05%. Okay. Now some important parameters is the degradation product of the level greater than reporting threshold should be summed and reported as the total degradation product. Impurities below reporting threshold can be disregarded. These are all ICH Q3B. If any unknown impurity is approaching to identification threshold, it needs to be characterized to control at the qualification threshold. Okay. Now, identification threshold. What is the guideline? Is again, it is starting from less than 1 mg or 1 mg to 10 mg, greater than 10 mg to 2 gram. These are the category based on the maximum daily dose. And if you see the identification threshold keep on changing there, what is the based on your the maximum daily dose. Here also I try to product one calculation. Say your maximum daily dose 300 mg for any drug product. So it will fall between 10 mg to 2 mg, this category, right? 10 mg to 2 mg. So to calculate, again, you have to calculate the simple arithmetic 2 by 300 into 100. It's 0.66%. So which is more than 0.2%. So what is the guideline say? Guidelines say if it is 0.2% or 2 mg of the TDI, whichever is lower. So in this case, it is again point limit should be 0.2%, right? Similarly, qualification threshold. Also, it is the category the same maximum daily dose and the qualification threshold. These are the category. These also I have given one example of hypothetical example. This is the hypothetical with respect and this thing. Once again, we have to calculate and we have to set what is the limit. Okay. So now come to the impurity specification. So whenever the impurity is concerned, what we have to know, this is the how to calculate the specification and what is the specification. It is in my opinion, it is based on the sound scientific information and the knowledge of the for the chemical reaction and conditions involved in the API synthesis steps, impurities associated with the raw materials and the intermediate. This understanding is very, very much important of the process chemistry, organic chemistry understanding to define the selection of the impurity. So how it will be part of specification, it depends on the batch manufactured and the proposed manufacturing process. And the rationale is also for the inclusion, exclusion, specification will be presented, because whenever you have to file anything, you have to present the rationale. What is the why you are keeping it in specification? Why don't you keep it in specification? Similarly, specified identified impurity should be included along with the specified unidentified impurities at the level greater than the identification threshold. Okay, these are the very brief summary, and I think if you read the guideline, much elaborations are given. Okay, then specifications that basically this is the same thing. I can skip this slide, and here is the some arithmetic. I can also skip this slide because of the thing. Okay. Now, impurity qualification. Impurity qualification is a very important thing. Qualification is the process of acquiring and evaluating data that establishes the biological safety of the impurity. And higher or lower thresholds for qualification can be appropriate on case-by-case basis. And scientific, rational, and toxicological studies data are very important. So, for example, qualification can be specifically important when there is evidence that such impurities in certain drugs or therapeutic classes have previously been associated with an adverse reaction. Basically, if I know some of the previous history from the literature or so, then I can qualify the drug as safe. Now comes to the ICH-Q3B mainly focuses on the degradation products of the drug products. So this is the different degradation product procedure. It may observe in the routine batch or the commercial batch. And a commercial batch, it may observe during the accelerated or long-term stability studies, or it is the then if what is important, the knowledge of the degradation pathway. So because we have to differentiate always the what is the process impurities and what is the degradation impurities, right? Now these are the types of the degradation products, and it could be once again in the same way. It could be specified degradation product. This could be unspecified degradation product. So specified degradation product, once again two categories: specified identified degradation product and specified unidentified degradation product. So in both the cases, your suppose it is a specified identified degradation product means when we are telling identified means that impurity we have done the all structural characterization. We have a thorough knowledge of that. And when it is unidentified, at that position means we do not have the much idea of the impurity structure here, but we are mainly moving by the some RRT. Suppose in a chromatogram with respect to it could be coelute with some known impurity, it could be something, but it is mainly by RRT or by using LCMS you can do the molecular weight. Okay. These are the types and important thing, because I think if all our specification queries, if you see the regulatory queries are, this is a very snapshot, but this understanding is very much required. Now the qualification of the degradation product. So whatever, how I will justify the qualification. Suppose when a qualification threshold are exceeded, the degradation product levels are to be qualified. So these are the process. You have to comparative analytical studies with the RLD. RLD is the innovator drug product. So from that we can take a decision that whether what is the impurity level of that same impurity level in that RLD. And again, suppose you have to take one another scientific and justification that it could be a metabolite or it could be whatever it is, or if we are not able to justify by these two, then the last stage is the toxicity studies, but it is not preferred because it is taking a lot of time and you have to prepare a lot of impurities also to do this. Because but even these first two is are very important if we can able to manage using these two things. Now come to the genotoxic impurity. So genotoxic impurities, or these are the class of class 1. It is basically from ICH M7 that class 1 is the known mutagenic carcinogens. This is guidance high control at or below compound specific acceptable limit. Class 2 is mutagenic but carcinogenicity is unknown. This should be same. Control guidance is same. Control at or below acceptable limit. Class 3 is the alert structure is unique and unknown mutagenic potential. Then also control at or below acceptable limit. Fourth category is if alert structure is non-unique and qualified in comparison to API, then you follow the Q3A or Q3B guidelines. And if there is no structural alert, in that case, these two are the same category. Now, these are the some very common functional groups, which are the genotoxic. First is the aromatic, these hydroxyaryls. Then in the alkyl and aryl groups, aldehydes and methanols, epoxides. These are all of a specific organic chemistry based thing. These type of functional groups or moieties are generally gives that N nitrosamine nitro compound. Nitrosamine became the different altogether different category nowadays. So these are all the structural. These functional groups give the structural alert. Okay, this is the thing. This is few I have mentioned, and a lot of things are there beside these also. Now, how we are evaluating the GTI? So there are some softwares that are there that direct, then Skyfeezer, Oncology, Toxtree, TopCat, all these things. These are the all toxicity prediction software. So all companies or different pharma companies are using this software to understand suppose the unknown impurity or any impurity, what is the structure alert is there. So after based on that, we have to decide what will be his fate. Now, limits. Once again, the how to set the limit. This is also the very specific guidance is there for class 1 control at below compound specific limit. So guidance is like here the guidance is based on the duration of the treatment. Okay. So suppose the treatment if you are taking for 1 month or 1 to 12 months, 1 year to 10 years, or one we call lifetime. Lifetime means 10 years to more. So the duration of the treatment means how long we are taking him. Suppose I know I am telling a very simple way. People are taking a hypertension drugs or say or the anything. Those are you have people are taking lifetime, right? Or maybe diabetic drugs like metformin, rosuvastatin, etc. So these are all category coming in the this category. Majorly these are this most of the thing it is lifetime category. So and one calculation again I have to show that is also how it is very well known that how to calculate the genotoxic impurity specification in parts per million. That what is the TTC in the microgram per day. That is suppose for it is a lifetime. If it is in this category, it is we have to consider 1.5 then divided by maximum daily dose. Suppose one raw material is a genotoxic in a product ABC with maximum daily dose is 0.02 gram. It is very simple 1.5 divided by 0.02. So it will come out 75 microgram per gram. So this is the specification. So it is very everybody knows. I am sure. And again, I am just repeating and to now it is the if it is the more than two impurities are there, class 2 or class 3 impurities, or then this is the basically everything is same. These numbers are different. If you go by the previous one, this is changing. So then you have to calculate accordingly, right? Now come to the this is little bit important the control of process related genotoxic impurities. Okay, there are four options is described in ICH M7. Option 1 suppose include a test for the impurity with an acceptance criteria. Option 1 stating at or below the acceptable limit in the drug substances specification. Option 2 at or below acceptable limit in the specification of the raw material, starting material, intermediate, or as in process control. See, in an API process, suppose this genotoxic impurity, I am controlling in the API stage with the whatever the calculation I have shown in that. That is just option 1. Or one organization can do I know the pathway of the formation of the impurity, so I am controlling at the raw material, starting intermediate or in process stages. That also I can do. In that case, I cannot, it may be skipped from the API specification. Option 3 is you can set it above the acceptable limit in the specification of raw material, intermediate, or in process. Now, suppose a product is A and B is the starting material, A, B is forming, C is the intermediate, C to D is the API. So, what this guidance is telling, you can do A, B, or in C stages, whatever the limit. Suppose last time, suppose it is 75 ppm, I am setting it at 150 ppm at that level. So, but in that case, you have to demonstrate the fate of the impurity must be below acceptable limit in drug substance. So these options are given. So if I demonstrate it with my development studies and the plant free exhibit batches data, there is no need to test it on batch on the every batches. So this is the purge study, etc. We have how to demo. Not normally people are doing it in the process control, purging or etc. Now option 4. Option 4, they have given the guidance given an option. You have to deep dive on the process parameters, including fate purge knowledge, with the sufficient confidence that the impurity in the drug substance will be below acceptable limit. No analytical testing is recommended. No need to list or data in any specification, but elements of scientific risk assessment should be prepared for justification. So these are the guidance. Say if you give a sufficient technical justifications on all these things, all development data, so no need to regular control is not required. But this is the guidance. But in a real sense, we are during filing we are showing all the data, and in many cases my experiences are mix and match. Sometimes agency accepted, sometimes agency say yes, you put it on specification or so, because once again the guide, the scientific justification, etc., it's a very broad and very great term. Suppose if the agency is not accepted with your, not very much convinced, they may say put it on specification. But in the guidance, these are there. So I believe in ICH M7, these option 1, 2, 3, 4 are very important from the development stage. When I am developing API and corresponding API to drug product, if I do, if organization do on a very in-depth, deep dive, in-depth level, then the chances of the regulatory queries are very, very less. But if we are not doing our homework on base studies at these stages very well, then there is no need to, there is no worry. You will get a lots of query. Again, you have to respond to the queries, and you will getting more time for that. You will losing time on that. So now come to the last part of the thing, the elemental impurity, which is ICHQ3D. This is also, so there are 3 categories, 4 categories: class 1, class 2A, class 2B, and class 3, and other elements also. So these are all well known. Class 1 is the big 4: arsenic, lead, cadmium, and mercury. And class 2A is the cobalt, nickel, vanadium. Basically, people are in the maybe the organic or analytical or the formulation, whoever this 7 names should be in the tips at any point of time. What is the elements? That's for with 4 and the 3. This is the compulsory you have to do. Class 2B are it is depend on your type of formulation, when this impurities to be included or not. Mainly for non-oral formulation, this is becoming important, any of this. And class 3 category is this, some barium, chromium, copper, lithium, molybdenum, antimony. And other elements are, apart from that, there are a lot of elements are there, which is like aluminum, boron, iron, calcium, potassium, manganese, sodium. These are, you have to, based on your uses in the process, you have to use it, but not specified under ICH category. Okay. Now, risk assessment process. This part is very important. This is very, what you have to know, you have to identify the known and potential sources of the elemental impurities. Evaluate the presence of the in particular elemental impurity in the drug product. and summarize and document the risk assessment document. These three are the most important stage of the risk assessment process. This is, I can skip this. This is the sources of the API from where. This is the metal, manufacturing equipment, processing aid, inorganic, organic material, solvent. These are all the sources of the elemental impurity in a drug substances. Similarly, potential sources in the drug product. Drug substance is the number one potential source. Apart from again manufacturing equipment, water, container closure system for the drug product, excipients. This is one of the major source. So for drug product, these are all the source. When you have to prepare the risk assessment, if we have to growth, we have to review all these data of the different vendors, excipient, and we have to prepare the risk assessment. I am trying to cover something. Sure. Then the recommendation for the elements of the risk assessment, this is very fixed. For oral drug product, there are seven. As I have told that seven should be people should know at any point of time. For parenteral, that seven additional class 3 lithium, antimony, and copper is there. Inhalation number is more, little bit more. So for oral, it is 7, parenteral 10, and total inhalation 14. So for this of the in your risk assessment, you have to compel or mandatorily you have to give justifications for all these elemental impurity, their sources, presence, control, XYZ. Apart from that, in your API manufacturing processes, if you intentionally add, suppose you are using, say, in API manufacturing process, a palladium catalyst, okay, nickel catalyst. So nickel is there. Suppose you are using a palladium catalyst, rhodium catalyst. So lot of catalysts are used in organic chemistry. So in that case, those elements you have to additionally you have to show the control. That is very valid for all these things. In formulation, what is the sources of the in formulation process? There are many milling system is there. People are very well known is the zirconium, what's called that some zirmil beads that are there. They are made up of zircon, mainly zirconium and etc. So in this, suppose if you are using in the formulation process those type of things, then you have to show the leaching of the zirconium, tungsten, etc. It also required. So mainly it is intentionally elements are last line. It is added from the API process only, but it is you cannot rule. Sometimes formulation process may also you have to consider. Now evaluation. Okay, so post impurity identification, two outcomes are possible. That one is the potential impurity identified, and another is the known potential impurities. If there is no potential impurities, then in any case, you have to show these 7 or 10 elements as a one-time data. The risk assessment report, this is very critical, and this is once again I have seen many a time we have made mistakes or mistakes been inadequate. I am not telling mistakes the right word. So risk assessment you should prepare it from the published literature, information of the supplier information. For I am mentioning this for formulation only, that prior knowledge, excipient data generation from other similar processes. So all type of information you should give in the risk assessment report to make this a very solid report. Otherwise, again, you may get off and on queries, unnecessary queries, and again you have to do some. You have the more the query, the you are increasing your corresponding analytical team's load. Okay, again, if you do prepare this that develop method, develop karo, validate karo, something like that. Okay? Now, risk assessment, critical risk assessment report. This is one risk assessment process, one of the report. Second is the total elemental impurity level from all sources in the drug product is expected to be consistently less than 30% of the permitted daily exposure. The additional control is not required. Okay. So, but when the variability of the level of elemental impurity should be factored, because variability is there, analytical, because these are very low level, analytical variations are also highly possible. So, analytical variation also you should be justified. And lastly, you have to prepare some 6 batches or sometimes 3 batches data before filing. Okay. Now, control of the elemental impurity, I am skipping this. This is basically process control. All these things are, I skip it. Now, come to a little bit converting. This is also, it is a very analytical or regulatory team's domain, that how to decide the converting PDE to concentration and specification. There are 2-3 options are explained in the ICHQ3. Option 1 is, first, you have to take your daily intake is not more than 10 grams. Okay. So if you suppose one example, I have copied it from the ICH 3 guideline only. Suppose that hypothetical drug product having 2.5 gram product. This is less than 10 gram, and these are the all excipients are there. Daily intake is this. Okay. Now, next, now from that I have this is the I am considering focusing on for calculation purpose all the 7 elements. Okay, and PDE of the 7 elements are fixed given in the ICH guideline. What is for cadmium? Say it is focus on cadmium for calculation purpose. It is fine. Now, then what is the his concentration? Because option 1 is we are considering it is 10 grams, so you have to divide 5 by 10, right? So it is 0.5 microgram. This is the concentration is 0.5 gram. So when you are making your drug product, if you are say this is a data, okay, permitted that same drug product, this is the drug substance MCC. Accordingly, this is divided by all the PDE divided by 10. These are the data. In this table, data is there. How you calculate the maximum daily intake? Very simple, concentration into 2.5. That is the your maximum. So this is what the maximum daily intake, right? So now 2.5 is the total dose. This is one part. If you see your product is qualified through this, there is no risk assessment is over. Technical part, arithmetic part is over. So no need to do further thing. Now, what is the option 2A? Option 2A is do not, only one difference. Do not consider 10 gram maximum daily dose. Put it the actual specific daily intake. So in this case, the same example is also from ICH E3D, 2.5 grams. So say focusing on say cadmium, you have to divide by 5 by 2.5. So 2 microgram per 2 BPM is the specification. In the earlier cases, it is 0.5 is the specification because it is 5 divided by 10. Here it is 5 divided by 2.5. Right? Equation is the same. This is the same equation even for the denotoxic impurity everywhere. The equation basic is same. So this is the equation. Now you come to option 2B. Option 2B is they are giving you a option. It is basically, if you come to the formula, basically each excipient and their maximum what is the PDE you have to calculate. And one of the basic differences between the option 2A and 2B is in the 2B it is giving you option that you can, because in the suppose I'm showing you the suppose one illustration, this is approach 1 and approach 2. This is your drug product, okay, and this is the daily intake, the same like the previous one. Now suppose you got, this is your measured concentration. Measured concentration means this is, you are the manufacturer of the drug product. This is the COA you got from MCC, lactose, calcium, all these excipient from different vendors, suppliers. Okay. So if this is the COA, now how we will calculate this? Very simple. 1.1 is the MCC daily intake. You multiply 1.1 into 0.1. Why you will multiply using this formula, right? So this is your total daily mass. And if you summed up all these things, what you will get? 1.2. So 1.2, this is the, I am showing this example for lead only. For each element, you have to do like that. So 1.2 is the total. So what is the lead PDE? Lead PDE is 5. Okay. So if 5, then 30% of PDE is 1.5. Right. 30%. So guidelines say if it is less than 30% of PD, you need not to check it. So it is 1.2. There is no need to do any analysis. Analysis means any specification exercise, right? But approach 2, this is the basically option 2B. Okay, approach 2B is, you see this is the API and this is the measured concentration. What is, you got 20 titanium, 20 iron oxide, 10. This is the specific measured oxide concentration. Is very low, iron oxide is very low. The option to be suggest you can change this specification, this concentration according to your whims and fancies, and you can make up lots of probable permutation combination. One of the cases I am showing here, see, we change the lead to the 0.5. This are you change, say calcium phosphate, you change it to 5. You do the titanium dioxide concentration. This is the specification. Basically, you are changing it. Why we are changing it? Then the question is why we are discussing this. This is all our hypothetical data. Suppose real-time data is titanium oxide, anything are closing to the specifications. So if you change it in a higher level, and you summed up the same using this same formula, 1.1 into 0.5 is 0.5, whatever. Here you are getting 4.98. Okay, that microgram. What is the total PD of lead? 5. So, but if you are giving the risk assessment using this approach, then what is the mandate for you? It is, yes, it is, you are passing less than 5, then but you have to put it on specification. This element, you can go this approach, but you have to go by basic, you have to put it on specification. But if you at least 30% less than PD, no need to check the specification. So once again, to summary, option 1 is your PD divided by the 10. Option 2A, PD divided by the actual maximum daily dose. Option 2B, you have to calculate each excipient with their each excipient's an API maximum daily dose and summed up. And if it is less than 30% PD, your job is over. No need to test it regularly. If it is more than 1.5, then you can use this type of permutation combinations. This is a lot of permutation combinations you can do. And if you are qualifying, but in that case, you have to put it on specification. So option 3 is nothing. Option 3 is using this option 1, 2A and 2B, you are not able to control, your products are failing or so. Then you have to do the actual analysis of all the elements in your specification, and if it is passing, you will pass, qualify. If you will send it to the market. If you are failing, that is failing. In that cases, suppose the formulation is like that, API and formulation combinations are like that, there is a probability of failing or passing. You have to basically change the process. If it is the old process or if it is a new process, you have to take care initially, because no one is doing the elemental impurities as a part of regular specification. Our everybody's intention target is this should be a one-time solid risk assessment and demonstration, so that there is no need to put it on specification, right? It is once again one illustration. Suppose one drug product strength is 100 mg. Say consider the lead as a element. Maximum daily dose of the drug product is say 1000 mg per day. So 1000 mg and the strength is 100 mg means how much unit you have to take the product? Is a tablet or capsule? 10 unit. Suppose 1 unit weight is 1000, then you have to calculate that it is a 10 gram per day. But if you see, it is a hypothetical. If it is, suppose this is a oral product, your limit will be 5 divided by 10, 0.5. And if it is a parental, in this case, it is same. Okay. But in many cases, oral and parental keep on changing if it is less than 10 gram or more than 10 gram. Okay, this is one simple example. And this is my last slide. I do not know. It is one question to the audience who are hearing. I am not telling that answer is right now, but similarly, suppose one drug product is 32 mg, maximum daily dose is 160 mg per day. Weight of 1 unit of drug product is 585. What will be the precipitation limit for cadmium using option 1 and option 2? Okay, this is the question. This is a very simple question. So you can answer this thing or you can, that is okay. Okay, this is, I think, from my side is okay, over. Thank you for all for listening.
All right. Thank you so much, Dr. Ganguly, for that detailed information and session on decoding the impurities. So now we will move ahead with the question and answer session. So we will take up questions which are there in the chat. So our first question is from Dharmendra Tiwari, Medley Pharmaceuticals. The question is, how can formulation and process change reduce the risk of nitrosamine formation?
Process team and reduce the nitrosamine. Okay, no issue. The nitrosamine, if it is observed in a sample, then from the formulation perspective, if you wanted to change, suppose if you say API, this is the nitrosamine. API team has worked and there is they are not able to reduce this particular level. The formulation team can use it with some sort of antioxidants like that is sodium metabisulfite and what is called vitamin C, calciferol. These are used, they can use to reduce in the formulation. In the formulation process also, suppose if it is the blister strip, etc., you can check from there also nitrosamine is come, possibility is there. Or API part, yes, API part is to, it is a, you have to control from the lot of the reagents, intermediates, and every step you have to understand the chemistry and you have to reduce the source of the nitrosamine. Say one famous and very great example of nitrosamine is the metformin drug products, right? So metformin drug products, it is dimethylamine, the starting material levels are, suppose if there is no control in the dimethylamine, suppose if you are controlling dimethylamine, residual dimethylamine in your API, there is a chance that you will not give the metformin in your drug, in metformin drug product, you will not get the NDMA type of impurity. This is one example I am telling you. Hope I am able to answer.
Dr. Ganguly, hi. Can you go back to the last slide where you had a question so that people can see that question and they can answer that? All right, audience. Once you have read the question, you can put your answers in the chat box so we can relay it to Dr. Ganguly and he'll get your answers.
No need to give the answer to me. It is just it is my nature. Whenever normally I put questions on the group.
Perfect. Then. We'll move on to the next question.
Okay.
We'll just take. I mean, I'll stop presenting your screen for you, and now you can see the next question being relayed on the screen, Dr. Ganguly.
I will. I think I have to escape
Okay.
this.
So
If you just go to the
the
browser, Dheeraj, can you just wait for a moment? Let
yeah,
Dr. come in the browser and see the question.
sure.
Can you
Where
see?
is
You can go to the Internet Explorer. E.
where you are telling? Otherwise, you tell the question. I am not able to see.
It's there in the. Okay, we'll read it out. No problem.
This is one. How do we specify or
Yes.
decide the
Yes.
limit of solvent and reagent which are not listed in ICH Q3C? This is also this is particularly solvents reagent which are not listed in ICH. This is the based on data, and here also some toxicity data uh, we are taking, not generating from the different literatures. We have to justify basically. There is no as such rule. You have to, which are not mentioned in the solvent guideline, you have to generate means from literatures only, that toxicity or all softwares you have to generate and say it is this is allowable or so. It is my understanding.
Okay, so our next question.
Yeah, I have seen
is-
the question.
Okay,
See, it is the in my experience, this they have accepting, but apart from that, that you have to show some data also, na, your actual data. I think they are accepting the toxicity prediction software based data. In my experience, they are accepting. What extent I am not very sure about the question, but I think if you say this is the toxicity data and this is the data observed in my product, and in many cases they are accepting, or in another cases is there. Many times they put no, they are. It is my experiences with nitrosamines are like that. Uh, I should not put the name. We put it to the go to the regulatory that this is the data. This is the our in-house toxicity team has generated. This is based on structure. This is the specification. FDA come back. Okay, if you propose something 1.25, we have proposed. Later on, they come back, say no, you can put it at 0.84 or something, right? So this is type of negotiations are going on. So there, but if you go that particular nitrosamine, what I am talking about, if you go by the 96 microgram per nanogram per gram, or but with that concept, the limit should have been 0.25. So, using Toxis software data, we have predicted and we have proposed 1.25, except FDA come back to a not, you should put 0.8. This type of things are there. But nowadays, this is I am talking about the for nitrosamine. This is the nowadays lot of after CPCA scores are there. You can predict the structure; they are acceptable.
okay, so we move on to the next question, and it's what are the requirements for vendor qualification in the context of impurity control?
What is the and little bit elaboration is required. Requirement for vendor qualification in the context of impurity control. What is the actual what is you actually wanted to mean? When vendor qualifications means impurity control, impurity specification is same for the vendor also. Suppose where the vendor means where the vendor means API vendor or the excipient vendor or raw material provider. Anyway, whatever it is,
Uh,
was
Dr. Ganguly, so this question is submitted anonymously. If you have further probe on that, I can also connect this particular delegate with you directly, because we have the option for the delegates to submit the question anonymously, as well as they can put it in the Q&A tab. So if
see,
you
I
have
am
want,
trying to, yeah, I understand. I try to answer because see, a vendor qualification means specific. If I am buying from a vendor, I will set up this is the specification it should be, because I will follow the guidance. So I will extend. Suppose I am a drug formulation manufacturer, I will expect API will follow all these ICH guidelines. If I am a API manufacturer who are giving the raw materials to us, So we'll put the specification. Suppose some of the specifications are higher, then in my process I have to control that impurity or their corresponding impurity in my drug substances to the below level. So accordingly, we have to discuss with the vendor and we say decide the specification. Similarly, for excipient vendor, suppose it is very common in nowadays. Suppose for nitroson, after the evolution of nitrosamine, I have to control the nitrite content, say in my excipient. So I will say vendor, this much nitrite content is not acceptable. You have to give me very low level of nitrite, and nowadays many vendors are giving it. So it is like that. It is a mutual discussion, and your process, matlab who is purchasing their process and their process capability and the regulatory guidance. You have to make a balance, or you have to do a trade-off between these two. I will say that is the right word.
Okay, thanks a lot, Dr. Ganguly, for that session. And we thank everyone for all these wonderful questions. And I hope we have concluded this really well. So, once again, we request everyone to give claps on your emoticons. Can we see that? Yes, wonderful. Uh, okay, just before we conclude, we have one last question. The
Okay.
question is impurities generated due to excipient interaction. Does these come under process or degradants? If the impurities are generated due to excipient interactions, these will be increasing in stability as well, I guess. I
Then
mean,
you have
yeah.
to control this impurity. This is a very common cases that they are lactose, Maillard reactions, etc. But these are not process impurity, because then you have to show the control of this impurity. This type of impurity you have to control in the specification. If it is this type of cases happening, because it will be,
Brilliant.
if
Okay.
it is increasing, no, if it is a reaction, suppose it is some lactose, mannitol, etc., some reactions are there, so control is required. But if you control it, or maybe you have to, some this type of cases are also there, you have to change little bit your manufacturing process also, for the formulation I am talking about. So but control is required. Specification based control is 100%.
Okay, wonderful. So, and thank you once again for taking up that one last question. And that was it. I guess we can once again thank you, Dr. Ganguly, for this. And let's again have a claps on the emoticons, please.